Hello, everyone. Good morning. Good morning, guys. How are you all doing? Good morning, everyone. Can I get good morning back from all of you? Yes, Mahima. Good morning. Good morning, Rajan. Good morning, Selva Priya. Good morning. Okay, everybody is doing fine, right? So here is a news for all of you. The CSIR NET paper, question paper has been released and the answer key has also been released. Now, in here also, in Biotechnica also, the experts here will be solving all of these questions. And soon we'll be releasing a whole webinar or video in which all the questions would be discussed with all of you. So stay tuned for that. Okay. Good morning, everyone. Yes. So yesterday's session, did you all enjoyed? And we also left in like 2 questions were being given to all of you. I hope all of you would have solved those questions. Yes. So today we'll be covering with the concepts related to Unit 3, where we'll be discussing about the major differences which are there between the prokaryotic and eukaryotic transcription mechanism, right? And there will be free sample papers also that you can download from the link in the description. At last, when the session gets completed, those free sample papers would be like provided to you, and you can fill the form and you can download them for yourself, right? So let's begin. So the topic of discussion today would be the difference between the prokaryotic and eukaryotic transcription, right? Now here we'll be comparing the fundamental processes of central dogma, that is the transcription, translation, and replication. What is the difference between them and how it is different both in prokaryotes and eukaryotes? And we'll be doing many comparisons actually, like what is the difference between eukaryotic transcription, how the chromatin structure of eukaryotes and prokaryotes differ. All these things, comparisons and explanation, we'll be doing with the help of Conceptica tools. We'll be taking help of concept tables and concept wheels. These are very much helpful, and it'll help you to remember these comparisons and the order in which the events takes place. For that also, these revision tools would be very helpful for all of you, right? So this is the major content of the discussion for this particular session today. So before going further with Unit 3, let us see, like you have solved the questions that I gave you for session, like yesterday, the session that happened for Unit 2. So these were the 2 questions that were being given to you. Let's see how many people have solved this. Yes, Amit, you have a question. Very good morning. Yes, influenza vaccine should be given every year. Okay. It should, due to the antigenic drift of the virus. Yeah, so CSIR paper, they set the paper according to the current events that are taking place. And so influenza virus, like every year, so they cannot give. Sometimes they give, and at that time you can attempt and you can give the answers, right? So yeah, these were the 2 questions that were being given. How many of you have tried and solving these questions? Did anybody tried it? One is the match the column, and other is the question related to the membrane transport. Which one of the following options is false? Yes, so Mahima said that simple diffusion fixed law. Okay. Okay, Mahima solved it. Amit Thakur, but CSR give answer regarding the incubation period, ma'am. Yes, so Amit, all these discussions we'll be doing at the last of this session, okay? Let me complete the whole session first, then I'll be giving answers to your queries, right? Selvapriya, you also solved? Okay. So let us begin. So match the following. Let us see like how the columns, both the columns, they match. So P class ATP pump first is, so which one of the in the second column, this particular, yes, ATP pump would be the example. And so P class ATP pump is being given here, and in the second column it is being given the energy from the hydrolysis of ATP and stored energy from ionic gradient, Fick's law, sodium potassium ATPase, and GLUT1 transporter. So as we know that sodium potassium ATPase is one of the example of P class ATP pump, right? So here is the right match for P class ATP pump. Then comes uniport transport. So I'm solving it. Those of you who have solved this earlier, you can check for yourself like if you're right or wrong. Okay. So for uniport transport, what is there? For uniport transport, we discussed the example was GLUT1 transport, right? Then comes the secondary active transport. So secondary active transport, it utilizes the energy of in the form of the ionic gradient, or we also call is the electrochemical gradient, which basically consists of the gradient, the concentration gradient of the solute plus the charge that is present, the variation in charge that is present across the plasma membrane. So voltage across the plasma membrane. So both of these combined is known as the electrochemical gradient, right? Or ionic gradient. So the energy is basically being stored in the form of this ionic gradient, which is being utilized by the secondary active transport. Simple diffusion, it follows the Fick's law, right? It's correct, Fick's law, because whichever molecule or whichever solute which directly interacts with the plasma membrane, all those the transport of all those molecules, it follows the Fick's law, and it basically happens in simple diffusion, right? For the primary active transport, the energy from the hydrolysis of ATP, directly the energy from the hydrolysis of ATP is being derived. So this is the correct answer for this. So this is the match the following, like this is how you should match all of your answers. So how many of you have done it right? Yes. Okay. So most of you have solved this. Now coming to the second question, which of the following statements regarding the membrane transport is false? So in these four statements, which of the following is incorrect? The first statement says that the polar and the charged solutes will not cross the cell membranes effectively without the specific carriers. This is absolutely correct, because polar and charged solutes, they are not able to pass directly through the plasma membrane. They require certain kind of transporters. So this statement is correct. Coming to the statement B, it says that each protein carrier will only bind and transport one or a few very similar type of solute. So this statement is also correct. The protein carriers are very much specific to which solute it will bind, and one solute at a time only they transport, one or two. Right. Then coming to the third statement, which says that the sugars such as glucose are always transported by active transport rather than by facilitated diffusion carriers. So how many people think that this statement would be correct? Yeah, option C, many people says yes, it is incorrect. So why it is incorrect? Because as we have noticed that the glucose, for the transfer of glucose, it requires it can be transferred through both active and facilitated diffusion. So here it won't be visible. Wait a second. Yeah. So as we know that glucose, it could be transferred by both active and facilitated diffusion, right? When from the gut to the epithelial cells of intestine, right? When the glucose has to transfer from gut to intestinal epithelial cells, then the requirement of ATP is required, the energy of ATP is required. So this is a kind of active transport. And facilitated, we have discussed yesterday that the example was GLUT. So GLUT1, it basically helps in the transfer of a glucose from outside the cell to the inside the cell. So this particular statement is incorrect. Last statement says that the ions are typically transported by special proteins that form the membrane channels. Yeah. So the correct option for this is option C. So whoever have marked the option C, they are correct, right? So let's proceed. Now comes, yes, comparison between the three fundamental processes in central dogma. Now, all of you know like what central dogma actually is. This is the basic of the molecular biology. So from DNA, RNA is being formed, and from RNA, what protein is being formed, right? This process is known as transcription, and this process is known as the translation, right? Now, in prokaryotes and eukaryotes, how these replication, transcription, translation processes, which are the fundamental processes of central dogma, they differ. So through this revision tool, we'll be understanding the basic concepts. So first is the replication. So in replication, DNA is the template molecule. So in replication, as we know that DNA would be the template molecule. For transcription also, DNA would be the template molecule from which the mRNA would be formed, right? For translation, RNA that is being produced, that the mature RNA that is being produced at last, when all the splicing and capping and all these post-transcription modification is being done, that modified RNA then would be used as a template and it would be translated into proteins, right? Then is the DNA is synthesized in the process. So in replication, DNA is being synthesized. In transcription, RNA is being synthesized, specifically mRNA, because that basically consists of the genetic code, which is further is being translated into the protein, right? In translation, the polypeptides are being synthesized in the process. This is general thing. Everybody knows it, right? So the DNA polymerase is the key enzyme in replication. In translation, RNA polymerase is the key enzyme. And in translation, larger subunit of the ribosome holds the catalytic ability. Now, here is a question for you. In replication, for both prokaryotes and eukaryotes, give me the names of different types of DNA polymerases and RNA polymerases also, both in eukaryotes and prokaryotes. So I would be inviting answers from you. Yes. For replication, both in eukaryotes and prokaryotes, what are the different types of DNA polymerase that are involved? And which of the DNA polymerase is majorly responsible for the replication of the DNA, both in prokaryotes and eukaryotes? Yes, this is a question. Yes. So Somaraya Bhattacharya, she says that polymerase 1, 2, 3, okay. DNA polymerase 2 is, okay. Prachi says that DNA polymerase 2 in prokaryotes is majorly responsible for, because DNA polymerase 2 is responsible for DNA repair. DNA polymerase 3 is the major enzyme which is responsible for the replication process. Yes. So Savari Mitra says that DNA polymerase 3 is major. Yes, you are right, Savari. Prachi, you need to correct. Mahima says DNA polymerase 3. Yes, you are correct. Okay. Yeah, for eukaryotes, Somaraya says, Bhattacharya says that it is alpha, beta, delta, and gamma. Right? Absolutely right. Okay, okay. So yeah. So let us see how it is. So in case of eukaryotes, there are basically alpha, beta, gamma in delta, and epsilon. These are the 5 major DNA polymerase enzymes that are involved, out of which epsilon, DNA polymerase epsilon is the one which is majorly responsible for the replication process, right? In case of prokaryotes, what happens? Everybody knows that prokaryotes have like 3 types of DNA polymerases, DNA polymerase 1, 2, and 3, out of which 3 is majorly responsible for the replication process, 2 is majorly responsible for the DNA repair, and 1 is basically used for the gap filling when the primer is being removed, right? So it is basically involved in gap filling. So so this is the major thing. These are the basic concepts, you need to remember it. Coming to the other differences, then comes that in replication, template is being read in 3' to 5' direction, right? In transcription also, the template is being read in 3' to 5' direction. While in case of translation, it is opposite. The template is being read in 5' to 3' direction. Now, when I'm reading all these sentences, you should know, like you should have an image in your head, like how the 3' end and 5' end there, like what is the direction of the synthesis of the DNA in case of replication, what is the direction of the synthesis of the mRNA in case of transcription, and how the template is being read, right? Then other differences starts at ori. So replication starts at the origin of replication, and transcription, it starts near the promoter region. Now, promoter region are very important in both in case of prokaryotes and eukaryotes. You should know the difference. What is the difference in the promoter regions in both of these cases, right? And in case of translation, the translation basically starts from the start codon. That is, yes, I would be knowing like what would be the start codon. This is very basic. All of you should know this. What would be the start codon, like general start codon in case of translation? Yes, it would be AUG. So Prachi said AUG, Sovri said AUG, yeah. So coming to the next explanation, it says that the prokaryotic replication occurs in cytoplasm. Now, location is also very much important. Where the replication is taking place, where the transcription is taking place, and where the translation is taking place, both in case of prokaryotes and eukaryotes. In case of prokaryotes, what happens is transcription, translation, both of these occurs in the cytoplasm itself, right? Like there is a coupling of transcription, translation takes place. When the transcription is being like going on, then only the translation starts in case of prokaryotes in cytoplasm. While in case of eukaryotes, they have a very complex structure. The DNA is very complex and it is not being accessible to the enzymes that frequently as compared to prokaryotes. So in them, what happens is, firstly, the transcription takes place inside the nucleus. Then for the translation, the mRNA which is being produced, the pre-mRNA which is being produced, it has to come out of the nucleus and has to travel to cytoplasm where the actual enzymes for translation are there, right? So location is very much important. So in prokaryotic replication, it occurs basically in cytoplasm. Prokaryotic transcription also occurs in cytoplasm, and prokaryotic translation, it also occurs in cytoplasm. For eukaryotes, the replication occurs in the nucleus, transcription occurs in the nucleus, and the translation also occurs in cytoplasm, right? Then coming to the last, that is the prokaryotic DNA replication termination. Here we are talking about the termination. Now, usually when the two forks meet and fuse, creating two separate DNA stranded DNA molecules. So what happens in case of prokaryotes? There is a catenated DNA is being formed, and when the replication terminates, these catenated DNAs, they separate from each other and produces two different DNA molecules, right? Then coming to the termination of the transcription. The termination of the prokaryotic transcription basically occurs by the extrinsic and intrinsic termination mechanisms, where you would have read about the row-dependent, row-independent kind of terminations. Those also are very important questions, are frequently being asked in CSIR NET exam related to what are the events in row-dependent, what is the difference between row-dependent, row-independent transcription, right? In translation, with the help of stop codons, the translation activity, it gets stopped. Now, like I want to ask you the question, like what would be the stop codons? There are basically three types of stop codons. I want to know what are they. So I would be expecting answers. Yes. So there are three stop codons that you need to answer. Yes. So Pranathi says that UAG, UAA, UGA. Yeah, Pranathi, you're absolutely correct. These are the three the stop codons that are responsible for the termination of the translation, right? Then we'll move further. Yes. Now comes the comparison between the prokaryotic and eukaryotic transcription. Now here the location plays the major role. Now from the figure itself, you can see that here is the DNA in the of the bacteria. So this DNA first gets replicated, and then RNA polymerase comes and transcribes this particular DNA. mRNA is being produced here, and when the transcription is taking place, at that very time only, the ribosomes, they assemble and they onto the mRNA, and they like produce the proteins. So translation and transcription, they are being coupled, and this activity is basically taking place in cytoplasm, right, in case of bacteria. In case of eukaryotes, since the eukaryotic DNA is very much complex, right, the DNA is not being it is the enzymes which are majorly involved in transcription translation, they are not able to act upon the DNA of the eukaryotes, because it is present in a very compact structure in the form of it is wrapped around the histone, and in the form of nucleosome, it is present inside the nucleus. So before transcription takes place, first of all, the transcription factors come and they help the enzymes, the RNA polymerase especially, so that it can act upon the DNA for the transcription process, right? So for the transcription, the pre-mRNA is first being formed. This is the precursor mRNA first that is being formed, and the transcription process basically happens in nucleus for the case of eukaryotes. So there is a pre-mRNA which is being formed from the 5' to 3' direction. After the pre-mRNA is being formed, further the co-transcriptional modification also takes place. Now, in case of prokaryotes, there was no co-transcriptional modification or post-transcriptional modification was not taking place, because the translation and transcription was occurring at the simultaneously, like at the same time. But in case of eukaryotes, what happens is the pre-mRNA molecule, it has to travel from the nucleus to the cytoplasm for further processing and for the translation to happen, because the translation enzymes are majorly present in the cytoplasm of the eukaryotes, the eukaryotic cell. So before going to the cytoplasm, what happens is certain kind of modification and processing take place in the pre-mRNA. These are like 5' capping, polyadenylation at the 3' end, and the splicing activity takes place. Now, why this post-transcriptional modifications are required? Now, when the pre-mRNA will move from the nucleus to the cytoplasm, what happens is there is the presence of certain exonuclease enzymes. So to keep the mRNA in its stable form, and so that the mRNA is intact, it doesn't get degraded through the exonuclease, so 5' capping is required and polyadenylation tail is being required. And there are certain non-coding regions which are present that are also being removed with the help of splicing. So all these processing is required in case of eukaryotes. So you can understand how complex the eukaryotic DNA and the mechanism, whole mechanism, how complex it is. So this is the basic overview of what is the difference between the eukaryotic and prokaryotic transcription translation activity. Now, one more thing is there, that when the mRNA is being produced from, in case of bacteria, what happens is the length of the mRNA, it remains same. Like whatever the DNA that is being transcripted, the mRNA also has the same length, like whatever the DNA length is there. But in case of eukaryotes, what happens? In eukaryotes, the pre-mRNA, which is being formed from the transcription from the DNA, the pre-mRNA, the length is quite big. The length is quite large as compared to the after the post-transcription modification, the mature mRNA which is being formed. So mature mRNA is of shorter length, right? So these are the basic things, but still, like, we forget these basic things also, right? Then coming to the other differences between the prokaryotic and eukaryotic chromatin structure. Now, as we know that in case of prokaryotes, the DNA is very much accessible for the enzymes. That is why the transcription translation takes place simultaneously in the cytoplasm. But in case of eukaryotes, the DNA is very, the length of the DNA is very huge. So to fit it inside the nucleus, it has to be compacted. So for the compaction, what happens is the DNA in eukaryotes, it gets wrapped around the histone proteins, right? So, and it forms the nucleosomes. So whenever the transcription is required, then only the few segment of the DNA, it will be provided, or it will be open for the enzymes for them to assist on them, right? So here comes the first major difference, that the DNA in prokaryotes is much more accessible to the RNA polymerase, right? While few other proteins are involved in the to provide the interaction between the eukaryote and DNA and RNA polymerase. So in case of eukaryotes, there are certain other proteins that are required for the RNA polymerase, for the DNA to be accessible for the RNA polymerase for its processing and for transcription to take place, right? In case of eukaryotes, the DNA is being wrapped around the proteins called the histones to form the structures called the nucleosome, right? And in case of eukaryotes, the DNA is being packed to form the chromatin structure. So it is present in a packed form. Then is the other major difference between the RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Now, questions in CSIR NET are asked from both of, like, all of these RNAs. Like, what is the major difference between all of these RNAs? What are their functions, right? So all these three RNAs are majorly present in case of eukaryotes. So first comes for RNA polymerase 1. So RNA polymerase 1 refers to the RNA polymerase that only transcribes ribosomal rRNA, right? So basically RNA polymerase 1, it synthesizes rRNA, right? In rRNA, you would be knowing that there is the 18S rRNA, there is 26S rRNA, and there is 5.8S rRNA. And there is one more present, that is 5S rRNA. But this particular RNA polymerase 1, it basically synthesizes these three subunits, only 18S rRNA, 28S rRNA, and the 5.8S rRNA only in case of, this is what it does. And 5S rRNA is being synthesized by some other RNA polymerase. We'll be knowing about it. Coming to RNA polymerase 2. So RNA polymerase 2 refers to the central enzyme that catalyzes the DNA directed by mRNA synthesis during the transcription of protein coding genes. So RNA polymerase 2 is the major enzyme. It is a very important enzyme which is majorly required for the synthesis of mRNA, right? Other than mRNA, it also transcribes the miRNA, the snRNAs, and the snORNAs, right? snRNAs are basically the short nuclear RNAs which are required during splicing. And snORNAs are basically the microRNAs which are required during the other activities. So these are the major functions of RNA polymerase 2. And RNA polymerase 2 considering the major enzyme because it is required for the transcription of mRNA, which is the part of the central dogma, right? mRNA would be synthesized always. Then coming to the RNA polymerase 3. So RNA polymerase 3, it basically does what? It transcribes the untranslated RNAs and the tRNAs. So RNA polymerase 3, remembering the function of RNA polymerase 3 is also important because it transcribes the tRNA, right? And other than tRNA, the 5S rRNA that we said that on RNA polymerase 1 was synthesizing rRNA, in that the 5S rRNA which was left, it is being synthesized by RNA polymerase 3. So all these activities are very much important. Questions are frequently being asked in CSIR NET related to what kind of RNAs are being synthesized by what kind of RNA polymerase, right? Then, yes, other important question which comes in exam is basically related to the alpha amanitin effect on these particular RNAs. So for RNA polymerase 1, alpha amanitin, it is insensitive. For case of RNA polymerase 2, RNA polymerase is sensitive to even 1 microgram per milliliter of alpha amanitin. So even small amount of alpha amanitin is present in the solution, the RNA polymerase activity would be hampered. For RNA polymerase 3, it requires a little bit more concentration of alpha aminotin for its activity to be like restricted. So this is less sensitive. This is very much sensitive, and the RNA polymerase is insensitive regarding alpha aminotin, right? Yes, Mahima. So here is the information for all of you. So SURE batch has been starting from 4th March, and it is the batch basically for the CSIR NET aspirants. It would be targeting the CSIR NET examination, the 2 CSIR NET examination which will be held in 2022. Now here will be various like assistance that would be provided to you. Like there will be raid classes by CSIR NET experts, interactive sessions would be there, doubt solving chats would be there 24/7. You'll be provided with the assistance, and the hard copy of the study material would be provided, and video backups will also be there. So all these things would be provided to you if you join the batch. So for further information, if you want to know more about the batch, you can call us into the toll-free number that is flashing on your screen, and you can also like text us, or you can also get information through mails, right? Yes. Now here is a very important discrimination between both prokaryotic promoter and eukaryotic promoter. The promoters play a very important role in case of transcription, right? So for prokaryotic promoter, what happens is the promoter is recognized by the RNA polymerase and an associated sigma factor, which enables the RNA polymerase to locate the promoter. Now, if I want to like ask you, what is the like RNA polymerase subunits of RNA polymerase in case of prokaryotes? Like how many subunits are present? This is a question. Yeah, here is a question for all of you. Like in case of prokaryotic transcription, there is a RNA polymerase which is involved. Only one RNA polymerase is there. Now, what are the different subunits of that particular RNA polymerase? Anybody? Any answers? Okay, Amit says, okay, small and large subunit. Sauri says, Amit says there are 5 subunits, right, Amit? Like, can you name the subunits? Sauri Mitra says 30S and 50S. I'm not speaking about ribosome here. I'm speaking about the RNA polymerase. What are the subunits of RNA polymerase? Alpha, beta, sigma, epsilon, gamma. Okay. Amit Kumar says alpha 2, alpha 1, 2, beta, beta dash. Yes. Yes, video zone. You're right. There is one more. Okay. Devanshi says 5 subunits. So what are these 5 subunits actually? So in case of prokaryotes, the RNA polymerase enzyme has 5 subunits. That is 2 alpha subunit, beta, beta dash, and gamma. Right? These are the 5 subunits. These are known as the core subunits. Apart from that, there is another subunit, that is the sigma subunit, which basically varies from one like organism to organism. So it varies, sigma subunit, and in sigma subunit also the most common one is sigma 70, which is mostly being studied and is present in E. coli, right? So these are the 6 subunits. The combination of these subunits is majorly known as the holoenzyme. Right. So until and unless this RNA polymerase is present, like both of these core subunits and the sigma subunit, they combine together and form the holoenzyme. Until and unless this particular whole enzyme is not being assisted upon the mRNA, the transcription won't initiate. Right. So sigma factor is majorly being involved. It is basically required. It helps the core subunit to act upon or to fall upon the promoter region. So to recognize for the recognition of the promoter region by the core subunits, this sigma subunit is being required, right? So in prokaryotic, that is what it is being said in the first definition. The promoter is recognized by the RNA polymerase and an associated sigma factor, which enables the RNA polymerase to locate the promoter, right? This is the role of the sigma factor. Second is the prokaryotic promoters are simple consisting of short consensus sequence where RNA polymerase binds. So there would be certain consensus sequence present in the promoter region. There is the Pribno box, which consists of the Tata box, where majorly the RNA polymerase it binds, and there is the TAGA-CA sequence is there, which is present as minus 35 upstream of the initiator region, right? So these sequences are very much conserved in all the organisms in case of prokaryotes, right? So you should remember the sequence of these consensus sequence, right? And then is the promoter contains two short sequences elements approximately 10 and 35 nucleotides upstream from the transcription start site. Now some promoters contain one or more upstream promoters elements. Now some promoters also can contain some kind of upstream promoter elements, right? These are known as the UP elements. So coming to the last one, it says that the sequence at minus 10 upstream, you should know like what is the what does minus means, negative sign means, what does the positive means. So negative is basically the upstream and positive is basically the downstream, and we are saying this with reference to the initiator region, that is the from where the transcription basically starts, the transcription start site, right? So minus 10 elements, so at minus 10 base pairs upstream from the initiator sequence, there would be a consensus sequence present that is known as the Tata box, right? Tata at. And the consensus sequence at minus 35 would be, the consensus sequence is TTGACA. So this sequence you should remember. Questions can come related to the sequence in exams, which could be very confusing if you don't remember what is the sequence, right? Then coming to the eukaryotic promoter. So in case of eukaryotic promoter, the process is more complicated. As we know that the eukaryotic DNA is very much complex, right? It is wrapped around histone in the form of a chromatin structure, which is very compact. It is present inside the nucleus. So it is not present for the enzymes to act upon it. Like RNA polymerase, it is not accessible. So for the accessibility, what happens is certain other kind of proteins are required. These proteins are basically known as the general transcription factors, right? So firstly, the general transcription factors will be binding on the promoter region. Then only the DNA would be like, the DNA would be accessible for the RNA polymerase. And further, then after that only the transcription initiation will start, elongation will start, and then it will terminate, right? The eukaryotic promoters are complex and may consist of core promoter. Now there are three different parts of eukaryotic promoter that you need to remember. First one is the core promoter, second is the proximal promoter, and third one is the distal promoter, right? Now, with the help of the figure that I'll be showing in your further slides, you'll be knowing what are the core promoters, what are the major components of the core promoters, what are the major components of the proximal promoters, and what are the major components of distal promoters, right? Then coming to the other definition, which says that the eukaryotic promoters are diverse and can be difficult to characterize. The Tata element and the BRE typically are located close to the transcription start site. So the two of the components of the core promoter. Now core promoter is something which is mandatory. It is required. Unless and until the core promoter is present, the transcription or the gene expression won't occur, right? Other than that, like proximal promoters in some of the genes, the proximal promoter and the distal promoter may or may not be required, right? So Tata element and the BRE element are the major components of the core promoter, right? transcription start site, typically within the 30 to 40 base pairs. So these Tata elements and BRE elements of the core promoter, they are majorly present at 30 to 40 base pairs upstream of the initiators or the transcription start site, right? Then coming to the other definition, it says that the eukaryotic RNA polymerase 2 dependent promoters can contain a Tata element consensus sequence, which is TAT-AAA. The sequence is basically, which is very much conserved throughout the organisms, which is recognized by the general transcription factor Tata binding protein. So TBP is very much important. And it basically recognizes this particular consensus sequence in the Tata element, right? And then what comes a B recognition element, which is known as the BRE. The full form of BRE is basically B recognition element, which is recognized by the general transcription factor TF2B. So when the transcription factors are coming one by one, so there comes a transcription factor 2B, which basically what happens is it basically binds to the BRE region of the core promoter, right? So all these things are very much important for you to learn and to understand what is the major difference in the prokaryotic and eukaryotic promoter region, right? Then coming to the what is the Pribno box. In case of prokaryotes, what happens? There is a Pribno box present in the promoter of the prokaryotes. It contains the consensus sequence Tata at of 6 nucleotides. So 6 nucleotides are present. The sequence is very much conserved and you need to remember this sequence. Then comes, it is centered roughly 10 base pairs upstream from the site of the initiation of the transcription, right? So it is present like at minus 10 base pair upstream of the initiator region, the transcription start site, we say it, right? Then coming to the holoenzyme RNA polymerase. So in holoenzyme that we discussed here. So in holoenzyme, what happens is, here is the core subunit present consisting of the 5 subunits and plus there is a sigma factor, right? So this holoenzyme basically recognizes and binds directly to this particular region of the promoter at Tata, like Tata box. Then what is Tata box? So in case of eukaryotes and archaea, the Tata box is basically being present. It contains a Tata box consisting of TAT-AAA sequence. The sequence is different here. So both of these sequences, you should know what is the sequence of the Pribno box and what is the sequence of the Tata box in case of eukaryotes and in case of prokaryotes respectively, right? Located at the 25 to 30 base pair upstream of the transcription start site. So this Tata box is basically present 25 to 35 base pairs upstream of the transcription start site. Then it says that the promoter recognition cannot be carried out by RNA polymerase alone. So RNA polymerase in case of eukaryotes is incapable of accessing this particular promoter region of the DNA, right, for the transcription to take place. So for the assistance, there is the requirement of certain other proteins that we call as general transcription factors, right? But auxiliary proteins in the cell should recognize the promoter. So auxiliary proteins here, it is talking about the transcription factors, thereby recruiting a specific RNA polymerase to the promoter. So this is what the basic definition of like comparison between both the eukaryotic and prokaryotic promoters is. And these differences are very much important. Though questions like may not be asked directly from the sequence of the promoter or something, but you should have a clear-cut idea of like what the promoters of prokaryotes and eukaryotes really look like. So through the help of diagram, it would be more clear to you. So here is the diagram. So for the prokaryotic promoter, what happens is the promoter region basically consists of, yes, the Tata at, this is the sequence, this is the consensus sequence which is present at minus 10 upstream of the transcription start site. So this is the transcription start site. So from here, minus 10 base pairs upstream, it is present, and minus 35 base pair upstream from the transcription start site, there is another consensus sequence which is present. This is known as the TTGACA sequence. So both of these sequences you should remember, right? So this is the transcribed region. And between both of these consensus sequence, the optimal inter-base distance is basically 17 base pairs. It could be plus minus 17, right? Then coming to the eukaryotic promoter. Now, eukaryotic promoter is quite complex, and as we discussed, there are 3 major parts of the eukaryotic promoters. First is the core promoter, And second is the proximal promoter, and third is the distal, that is the regulatory elements are present here. So core promoter, as we know, the components of the core promoter are, there would be the Tata box which is present, right? This is very important, it is mandatory. There is a BRE region which is there present. So here basically the TF2B, right? This is one of the general transcription factor. This basically binds here. Then there would be the DCE region would be there, and DPE region would be there, which would be present downstream of the core promoter, right? So these are the three parts of the eukaryotic promoter which is very much required. Now this proximal promoter, from the name itself, proximal, it says that it is present nearby the core promoter, right? And for the distal regulatory elements, these can be required or cannot be required. These are the regulatory elements which are present. These basically involves the enhancers, there is the insulator present, and the silencer is present. These basically regulates during the gene expression, right? So DC is basically yes, it is the downstream core promoter. So this is the basic general figure. In the figure form, I have explained how the prokaryotic promoter and eukaryotic promoter differ from each other, right? Basic idea it is giving. So here is a question for all of you. So whatever discussion we had till now, there are two questions based on that. Now, I want you to solve these questions and tell me what would be the correct answer. Amit Kumar says, yeah, sorry, sigma. Okay, Amit, no problem. Yes, okay, fine. Here are the two questions that are being given and that are flashing on your screen. Can you please answer me what would be the correct answer for both of these questions? We have discussed this just now about regarding the sigma subunit, and it is regarding the alpha amanitin inhibition, right? Yes, anybody? Yes, so Mahima says about the second question, she says that alpha amanitin inhibits RNA polymerase 1. Okay, you said RNA polymerase 1. Any other answer? Sobri says C, RNA polymerase 3. For which question you are saying, Sobri? There are two questions here. Which question you are speaking about? Okay, for question A, everybody is saying option A. Millennial. He says that option C is the correct one. Okay. Any other? For question 1, option C, and for question 2, option D. So Sobri says for question 1, C, and for question 2, it's D. So let me tell you what would be the correct answer for this. Right? So first question says that the sigma subunit of E. coli RNA polymerase does not. Firstly, it initiates the transcription and fall off during the elongation. So this particular statement is correct, because sigma factor is majorly being required for the initiation of the transcription, and when the transition occurs from the transcription to elongation, then the sigma subunit, it falls off the DNA, right? So this statement is correct. Second says that there is increase in affinity of the core enzyme to the promoter. So sigma factor, another major function of the sigma factor is that it increases the affinity of the core enzyme to the promoter. So this option is also correct. Third says that the binds to the DNA independent of the core enzyme, right? Now, until and unless the whole holo enzyme is not being present here, holo enzyme, which consists of the 5 subunits plus the sigma factor. unless until all of like this enzyme RNA polymerase is not present in its holoenzyme form, the transcription activity won't start. So this particular option C, is the correct answer for this particular question, right? It does not binds the DNA independent of the core enzyme. It requires the core enzyme also. D says the ensures specificity of the transcription by interacting with the core promoter, core enzymes, right? So for the core enzymes to bind to the DNA promoter region, for its specificity, sigma factor is required. So whosoever gave the option C as the answer, they are correct. Yes. So Mahima says, okay, now Mahima, you are right, that alpha amanitin inhibits only RNA polymerase 2, right? So in option D, it is given all of the RNA polymerase. Now, all of the RNA polymerase cannot happen because RNA polymerase 1 is insensitive, right? It is insensitive, right? Just now we read it. But RNA polymerase is very much sensitive even if small amount of alpha amanitin is present to the solution, right? For 3, it requires little bit more amount, but RNA polymerase is the major enzyme. Polymerase 2 is the major enzyme for which alpha amanitin for which it is sensitive to alpha amanitin, right? So option B would be correct. Then coming to the transcription factors in their order and function. Now the order of the transcription factor is very much important. For this, you can use mnemonics. Like for me, like personally speaking, what I use is basically is DAB, FEH, right? So D is basically for TF2D. A is basically for TF2A, 2A. So in this order, I remember like what would be the order of the transcription factors which will be coming upon the promoter region so that the RNA polymerase can do its activity. So here with the help of the Conceptica wheel, you can see the order in which the transcription factors, they basically come onto the promoter region and assist the RNA polymerase for its activity for the transcription of the DNA to take place, right? So firstly, the TF2D is the one which comes, right? Now, the special factor about TF2D is that TF2D enzyme protein, it basically, the TVP region is there. The tita binding protein is there, right? It is the part of this TF2D transcription factor, right? And there are other subunits also present here. Multi-subunit. This is a protein which consists of multi-subunits, right? So there would be multi-subunits which would be present here, right? These multi-subunits are basically known as the TAFs. So tata binding assisting factors are there. So whenever in the core promoter, whenever the tata box is there, right? If the tata box is there, then only it will bind to the TBP, tata binding protein. If in the promoter, other than tata box, other type of sequence is present, for them, the TAFs, they come and they bind to that promoter region, right? This is the basic role. And like this is what is being explained about the TF2D. It is a multi-subunit. It consists, the TBP is basically present inside the it is a part of the, TF2D, right? And TBP is the major protein which is involved in binding to the tata region, right? Then comes TF2B, binds to the TBP, recruits the pol2. Okay, so the order that I told was, first of all, the TF2D will come, right? Which consists of the TBP region. This TBP tata binding protein will first bind to the tata box, right? After the TF2D has bound to the tata box, then comes the recruitment of TF2A. Right. After TF2A comes TF2B. Right. This TF2B further helps in the recruitment of TF2F. Right. So nomenclature, you would be knowing that TF is basically for the transcription factor and 2 is basically for RNA polymerase, and D is the order of the transcription factors with which order they are coming, right? So TF2F comes. After TF2F comes, it basically recruits TF2E, right? This TF2E further recruits TF2H. Now, TF2H is one of the very important transcription factor. It is having two major activities. So, TF2H has two major activities. One is the helicase activity, and another is the phosphorylation or the kinase activity. It basically says kinase activity is there, right? So first activity is the helicase activity. So TF2H is majorly involved in unwinding of the DNA for the transcription to majorly take place, right? Secondly is the kinase activity. In kinase activity, what does TF2H does is it basically helps the function. One of the major function of TF2F is it basically binds tightly to the polymerase. So RNA polymerase itself is not able to recognize the promoter region. So for that, help is being required from outside, and this help is being provided by the TF2F, right? After TF2F, TF2E is being recruited, and TF2E basically recruits TF2H. So TF2H having two major activities. One is the helicase, another is the kinase activity. Helicase activity, in helicase activity, TF2H, what does it do? It unwinds the DNA for the transcription to take place. And in kinase activity, what does it do? It basically phosphorylates the CTD tail of the RNA polymerase. This is very important. CTD tail is basically the carboxy terminal tail of the RNA polymerase. Until and unless this CTD tail is being phosphorylated, the initiation of the transcription won't occur, right? So for this, the TF2H is required. So TF2H is a very important transcription factor which is required for two major activities. Now here in the diagram, you can see that all the transcription factors have bound to the promoter region. RNA polymerase 2 is also there, which is bound. It is having a long tail. This is known as the CTT tail. That is the carboxyl terminal domain of the RNA polymerase. Now here what happens, this CTD basically consists of 52 repeats of heptamer of amino acids. Like there would be 7 amino acid sequence which would be present, which would be repeated 52 times in the CTT tail. So these subunits are basically, so here would be Y, S, P, T, S, P, S. So these are the 7 subunits that are involved. So Y is basically tyrosine, S is serine, P is proline, T is threonine, S is serine, P is proline, and S is serine again. So this is the 7 heptamer sequence of amino acid which is present in the CTD region, which is very much consensus sequence, and it is present 52 times in the CTD region. Now at the 5th position, the 5th serine, basically the phosphorylation takes place. So here in the second figure, you can see there is a tail, and at the 5th serine position, this is the serine, the phosphorylation majorly takes place, right? So what is the importance of this CTD? Firstly, when the CTD gets phosphorylated, then only the initiation starts. You can see that when the CTD region, it gets phosphorylated at the 5th position of the serine, then only this RNA polymerase, it has crossed the initiation, the start site, and the initiation complex have after the initiation complex has formed, the initiation has begun, right? So this is the first major function of CTD. Second major function of CTD is that, as we all know, that the pre-mRNA, before it gets converted into the mature RNA, it is required certain processing, right? These kind of post-transcription modifications are required. So the enzymes which are involved in the post-transcription modification, those enzymes are also bound to this CTD region. So these are the two major functions of CTD region, and you should also remember what is the heptamer sequence of amino acid which is present and at which position the phosphorylation is majorly taking place, right? So on that basis, whatever we discuss, this is a question that I have put up here, which says that the HeLa cell extract was used to study transcription of a gene X having 6 introns. RNA polymerase II complex containing all associated proteins was isolated from actively transcribing system and subjected to proteome analysis. Now, results showed that the presence of both splicing and capping enzymes in the complex. Now, the complex which was being formed, it says that both splicing and capping enzyme was present there. When the transcription elongation was inhibited by a drug, that is the flavopyridol, then what happens is the polymerase complex contained only capping enzymes, right? Only capping enzyme was present, the splicing part was removed. When the phosphorylation of the CTD domain of the Pol2 was inhibited, just the CTD domain that I was speaking about, that domain, the phosphorylation was being inhibited, right? By the kinase inhibitor, then what happens? The complex contains neither the splicing region nor the capping enzymes. Now you have to tell from these results, following conclusions were being made. First is that the transcription of the gene X is coupled to mRNA capping. Second is the transcription elongation is coupled to splicing. Third is the phosphorylation of the CTD is required for the recruitment of capping and splicing enzymes. And last option is both capping and splicing of mRNA occurs simultaneously. Now, what would be the correct statement according to the experiments which they have given here and whatever I explained till now about the CTD region? So this question is a bit tricky, but if you would have listened to me what I was speaking till now about the CTD region, you would be knowing like what would be the correct answer. You can give it. Okay. Smart Buzz, he says B option. Keetana says D. Okay. Different answers are coming. Dilshad, he says answer is A. Okay. Mahima says option is D. Right. Okay, Sabri says answer is A. Anupam says answer is D. Okay. So whosoever gave, like most of the people you are giving the wrong answer for this, but whosoever says the answer is A, they are right. Now let me explain you how the option A is the correct one, right? So firstly, it says that the RNA polymerase II complex containing all associated proteins was isolated from actively transcribing system. So the system is actively transcribing at this particular point of time and subjected to proteome analysis, right? So results, what it showed, that there is a presence of both splicing and capping enzymes in the complex, right? So as I've discussed that here is the RNA polymerase, sorry. So here is the RNA polymerase is there, right? It would have the CTD tail. It would be binding to the DNA and mRNA would be generated from here, right? 5' to 3'. Now this CTD tail, it would be consisting of the enzymes that are involved in post-transcriptional modification, right? Since this particular system is actively transcribing, so during the transcription process only the post-transcriptional modification enzymes, they will be coming and they would be binding to CTD region, right? So post-transcriptional modification enzymes consisting of both splicing enzymes, capping enzymes, and polyadenylation tail, that enzyme also would be there. So when the transcription elongation was inhibited by the flavopyridol, so transcription elongation was being inhibited, right? Initiation took place and elongation when it started, then it was being inhibited, the transcription was being inhibited by the drug, right? At that particular point of time, what was the result of what was observed was there was only the capping enzyme that was being present, which means that during the initiation process only, the capping enzymes, they come and they bind to the CTD region, right? So the first option says the transcription of gene X is coupled to mRNA capping. So this particular option is correct. Then it says that when the phosphorylation of the CTD domain of the POL2 was inhibited, when the entire phosphorylation of the CTD domain was being inhibited, then what happened? Neither the capping enzymes nor the splicing enzymes were involved, were present, which says that the phosphorylation, the R option, which says that the phosphorylation of CTD is required for the recruitment of both capping and splicing enzyme, right? So this option is also correct. Option P is correct, option R is correct. Now coming to option Q, what does it says? It says that the transcription elongation is coupled to splicing. So elongation is coupled to splicing while initiation is coupled to mRNA capping, right? So both of these three options are correct. Now option C says that both capping and splicing of mRNA occurs simultaneously. No, this is not the correct option because capping occurs, capping is a process which occurs co-transcriptionally, while splicing occurs post-transcriptionally. When the transcription has finished, then only the splicing takes place, right? So whosoever gave the option A as the correct option, they are right. So did you understood like how the explanation for this particular question is? Give me a thumbs up if you've understood, and if you want me to repeat also, you can say it. Okay, Mahima say it's clear now. Okay. You're welcome, Mahima. Okay, Devanshi also, it's clear, right? Okay, fine. So all of you are clear with the explanation, right? So then coming to the RNA processing in eukaryotes. So this is the very important event which takes place in eukaryotes, which majorly doesn't occur in case of prokaryotes. The answer, the explanation for this is basically the location. So in prokaryotes, the DNA is very much like, it's simple DNA and it's accessible for the enzymes RNA polymerase to act upon the DNA of the prokaryotes. And the transcription and translation both occurs in cytoplasm simultaneously in case of prokaryotes. But in case of eukaryotes, the DNA is very much complex and it is present in a very compact structure and it is not accessible for the enzymes such as RNA polymerase to act upon it directly. So RNA processing is being required there. What happens is pre-mRNA is being formed in the nucleus, and from the nucleus for the translation process in the proteins to be formed, it has to come out of the nucleus and go to the cytoplasm. And for going from nucleus to the cytoplasm, there is certain kind of processing which is being required for the pre-mRNA, because there would be exonuclease enzymes which will be present in the cytoplasm, which will act upon this pre-mRNA and it will cleave them. So the whole mRNA will get degraded. So to protect and to keep the mRNA stable, the processing is being required, right? So there are three major post-transcription modifications which takes place. First is the 5' capping, right? 5' capping is basically a co-transcriptional modification. Then comes the 3' polyadenylation. Poly-A tail is being it is being joined at the 3' end of the mRNA. This is the post-transcription modification. And then comes the splicing, right? All of these are the transcription processing activity which takes place at the pre-mRNA. So here in the structure, it's given that there is a DNA, there is an RNA coding sequence, promoter is being given here. So first what happens is the transcription by the RNA polymerase 2 addition of the 5' cap takes place at the 5' end. There is a cap. After that is the 5' UTR, that is the untranslated region. So 5' capping takes place at the 5' end of this particular pre-mRNA, right? Addition of the 5' cap when it happens, it basically happens when 20 to 30 nucleotides of the pre-mRNA is already being made. After that, addition of the 3' poly A tail takes place at the 3' end of this particular pre-mRNA. So pre-mRNA, what all things you can observe here? At the 5' end, there is a cap present. Then there is an exon region, there is an intron region, right? So introns are basically the non-coding region which are to be removed. So for that, the splicing activity basically takes place, right? The introns are being removed and all the exons are being joined together. Then is the 3' poly A tail that is being formed at the 3' end, right? So this is the mature mRNA which is being formed after the processing has occurred. So this particular mRNA, mature mRNA, the length of this mature mRNA would be quite small as compared to the pre-mRNA which is being formed because introns are basically being removed, right? Then after that, the translation takes place in the cytoplasm and the protein is being formed. Right. So the first comes the RNA capping. So function of the enzyme and the factors which comes in order. So in this, you need to remember about each and every enzymes which are involved in the RNA capping, right? So what do you actually mean by RNA capping? So in RNA capping, what basically happens is 7-methyl guanosine triphosphate, right? So addition of 7-methyl guanosine triphosphate at the 5th position or the 5th carbon of the first nucleotide of the mRNA, it basically takes place in RNA capping, right? So firstly, the enzyme which is involved is RNA triphosphatase, which basically removes the gamma phosphate from the 5' end, right? So there would be the 5' end of the pre-mRNA from which the gamma there would be how many phosphate groups would be there, right, attached like this. So this would be alpha, this would be beta, this would be gamma. So gamma phosphate would be first removed, and this activity is majorly being done by the RNA triphosphatase, right? After that, guanylyl transferase, it is the enzyme which what it does, it brings the GTP from outside and adds it to the terminal bisphosphate, the bisphosphate which is being which is remaining now at that terminal at the 5' end. GTP is being brought up by the guanylyl transferase and it is being added to this bisphosphate, creating a 5' triphosphate linkage. So this linkage is very much important. So this is a kind of very peculiar kind of phosphodiester bond. It is not like the regular phosphodiester bonds which we used to study. Regular phosphodiester bonds used to be from 5' end to the 3' end, right? 5' to 3' phosphodiester bond we have studied till now. But this is basically the 5' phosphodiester bond is being formed here, right? Why this kind of unique kind of phosphodiester linkage is being required? Because this kind of linkage is not being recognized by the exonuclease present in the cytoplasm and it cannot cleave the 5' end. As a result, the stability of the mRNA is intact there, right? So first enzyme is the RNA triphosphatase. Second enzyme is the guanylyl transferase, which basically transfers the GTP from outside. After that, the other enzyme is the methyl transferases. So at the 7th nitrogen of the guanosine, which is being which is attaching to the 5th terminal, to the 7th nitrogen of this guanosine, a methyl group is being attached. With the help of the methyl transferase, 7 nitrogen of the guanosine is being methylated, right? After that, what happens is the S-adenosyl thionine donates a methyl group. So the methyl group which is being coming, the methyl transferases basically transfers the methyl to the 7th position of the guanosine, but the methyl group which it basically comes from this particular molecule, the S-adenosyl L thionine it basically donates the methyl group, right? So this is the order of the enzymes which is basically taking place in case of RNA capping. What is the importance of this peculiar 5' phosphodiester bond that you should remember, and the order you need to remember, right? And what is the purpose of basically RNA capping? Then coming to the other post-transcription modification, that is polyadenylation. So in polyadenylation, this has also the same activity as if we speak about the 5' capping, right? So it also protects the pre-mRNA and keeps the structure intact so that it is not being cleaved by the exonuclease enzyme, right? So it basically protects the 3' end of the pre-mRNA. So in here also you need to remember the order of the enzymes, with which order they basically come. So firstly, what happens is the CPSF, that is the cleavage and polyadenylation specificity factor. It basically binds to the upstream region, which is basically the signal region, we say it, that is AAU-AAA. This is the poly A signal sequence which is present at the 5' end. So you can see here in the diagram that the CPSF, it basically binds to this region. So here is basically, let me change the color. So here basically it binds, and this is the region which is the poly A signal sequence is present here. So this is the signal sequence. The sequence you need to remember, right? After that, CSTF. So there is the CSTF which basically cleaves the stimulatory factor. Its name is basically the cleavage stimulatory factor F, which basically interacts with the downstream GU sequence. So downstream of this signal sequence, there would be a GU sequence which would be present here, with which this CSTF would be interacting, right? After that, what happens? The RNA cleavage factors are being recruited, CF1 and CF2, at which point this CSTF and CSTF, it gets removed. CPSF is still bound there. Then there comes the cleavage factor, that is the poly A polymerase enzyme. It basically comes, and it basically, what is its activity? It polyadenylates at the 3' end, right? Then comes the poly A binding protein. So when this poly A tail is being formed by the poly A polymerase, after that the poly A binding protein comes, right? PAB, it is known as PAB. This basically comes. This what does it do? Where the poly A tail is being formed, at that particular point, it basically binds itself. There are multiple proteins that come. So in the form of multi protein, it binds at the poly A tail. Its major role is that it basically makes the pre-mRNA into the circular form. So 5' end and the 3' tail, it is being circularized by this particular enzyme. Why this circularization is required? Because this is another form through which these activity basically prevents the degradation of mRNA, because exonuclease, it cannot cleave like within the DNA, right? Within the mRNA, it has to cleave at the terminals. So for that purpose only, it is being circularized, right? So this is about the polyadenylation. So both of these activities I have explained. Okay, Savri says the polyadenylation tail addition is basically a co-transcriptional or post-transcriptional process. So Savri, it is a post-transcriptional process. It's not a co-transcription process. Only 5' capping is the co-transcription process, right? So I would be clear for that. Now, coming to this particular question. This is related to the polyadenylation that we discussed in the earlier slide, right? This particular question is your homework. I would be discussing it tomorrow, right? Tomorrow also at 11 o'clock only we'll be meeting, and I'll be coming up with the 4th unit, and there only we'll be discussing this particular question, right? So this is a homework for all of you. So I hope you would have understood from this session. It would be helpful for you to building your concepts regarding the transcription process which takes place in prokaryotes and eukaryotes, how it is different, right? So thank you very much. And in the end, you can also download the free sample papers related to this unit from the description, that from the link that is given in the description, right? Thank you very much.